Hello, everyone, and welcome to today's webinar titled Utilization of the Luminex Platform to Discover and Develop Dual Toll-like Receptor 2 and 9 Antagonists for the Treatment of Neurodegenerative Diseases. This webinar is part of the 10th Annual Neuroscience Virtual Conference. I'm Antonina Salcido of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Luminex, a DiaSorin company. For more information about Luminex, please go to luminexcorp.com. Now let's get started. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find that live chat located at the right of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into that Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in your abstract window below the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Deanna L. Price, Executive Director of Biosciences at Neuropore Therapies, Inc. For a complete biography on our speaker, please visit the Biography tab at the left of your screen. Dr. Price, you may now begin your presentation.
Thank you, and thank you for that introduction. Before I get started today, I want to thank members of the Luminex Corporation team for the invitation, for sponsoring, and to LabRoots for organizing this event. The program data I will be presenting today represents the combined efforts of past and current members of Neuropore's Drug Discovery and Development team. The program was jump-started by a grant from Michael J. Fox to build an in vitro screening platform, followed by add-on funding to evaluate a promising program lead in an in vivo model of Parkinson's disease. And we've now leveraged on the developed in vitro and in vivo platforms to extend our evaluations to additional models of neurodegeneration. Now, Neuropore is a pretty small company, and so in order to facilitate our efforts on this program and other programs, we have leveraged upon a preferred network of collaborations with industry and academic partners, and I've shown a small number of those here. So the development of CNS disease-modifying therapeutics from concept to approval takes a great deal of time, effort, and money. Over 85% of combined phase 2 and 3 CNS-targeted therapeutics fail, and this includes both symptomatic and disease-modifying therapeutics. And I say that because of the approvals in CNS, very, very few have been disease-modifying therapeutics. And those are therapeutics that either slow, stop, or reverse the progression of a disease. So there have been some notable exceptions, primarily in multiple sclerosis, ALS, and more recently for Alzheimer's disease. So how can we improve our success rate? What can we do to improve the preclinical to clinical translation? And I'd like to briefly focus on two key translational challenges to developing disease-modifying therapeutics for neurodegenerative diseases, centered on the patient population and biomarkers. So first key challenge being centered around the patient population, where variability in clinical trial data can be largely explained by patient population-related factors. First, there are knowledge gaps in the understandings of all of these disorders with regards to mechanism, and there are usually more than one, the progression of these underlying mechanisms, and optimal points for intervention. And heterogeneity within clinical cohorts contribute to variability and ultimately to trial failure, including time. If you think of a patient population, you've got all these patients within your trial, and they have different times from a symptom onset, time from diagnosis, the rate of progression, their symptom profiles, and sometimes they even have, within the population, you have patients who are considered non-responders to your therapeutic intervention. So it would be great to know who they are before you start your clinical trial. On the right, I'm showing a schematic which is related to Parkinson's disease with time-dependent progressions of pathology overlaid with progressive symptoms. So briefly walking through the layered aspects from top to bottom, and this is just a small part of the complexity of Parkinson's disease. First of all, there are progressive accumulations of a protein called alpha-synuclein in the brain, and it turns out in the periphery as well. And this progressive pathology is associated with progressive changes in neurochemistry, neuronal cell death, autonomic dysfunction, neuropsychiatric features, and motor dysfunction. And all of our understanding of all of the above is complicated by additional complex and dynamic changes in autophagy and immune pathways, elements of those pathways, and the function of those pathways, as well as changes in network connectivity and dysfunction, and changes in cellular metabolism. Now, the challenges related to the patient population are related to the second clinical development challenge of biomarker discovery, development, and use. Now, there are different types of biomarkers, including diagnostic, patient stratification, target engagement, disease progression, and therapeutic response. Now, along the bottom of the slide are some examples of Parkinson's disease imaging, functional, and multi-omic biomarkers. Now, some of these biomarkers are potentially multi-purpose, but most are not. For example, the dopamine transporter PET imaging is used as a Parkinson's disease diagnostic test. However, it is likely not useful as a therapeutic response biomarker. A successful trial in Parkinson's or other will likely require multiple biomarkers. For example, a positive DAT scan could be an inclusion criteria biomarker for patient stratification with additional biomarkers for clinical symptoms such as the rating scale for Parkinson's disease, UPDRS, and target engagement biomarkers that are specific for your therapeutic intervention. So one emerging strategy is to utilize a combination of biomarkers and look for consistent patterns of multiple endpoints in early clinical trials. And using this approach, various biomarkers can be critically evaluated for sensitivity, selectivity, and robustness, and inform your clinical development program. So Neuropore Therapies develops small molecule therapeutics that target common mechanisms which drive neurodegeneration. Now, on the left is a schematic showing 3 key pathologies common to most neurodegenerative diseases of protein pathology, increased inflammation, and deficits in clearance mechanisms, which collectively we call autophagy. Now, many neurodegenerative disorders are associated with one or more abnormal aggregated proteins. They have markers for increased inflammation, and there are different types of autophagy that we have evidence for changes in and dysregulation. So Neuropore Therapies is pursuing 3 distinct therapeutic approaches based on this triad to develop therapeutics for slowing progression of neurodegenerative diseases by either directly interacting with toxic proteins, by inhibiting brain inflammatory processes involved in the disease, or by rectifying deficits in cellular mechanisms for clearing aggregates or misfolded proteins. I've led in vivo pharmacology efforts at Neuropore Therapies, along with translational science aspects of the programs. And the 3 key aspects of our in vivo pharmacology program are shown here. The first is a general approach and utilize technologies. We've chosen to invest in and use technologies that can be used regardless of discovery program or indication, so they're program and indication agnostic. We are a very small company with comparatively fewer resources, space, and time compared to larger companies, and so we have necessarily built a multimodal and multiplexed unbiased toolkit of study measures that are flexible, efficient, and support all of our programs. And here are some examples underneath of some types of those data evaluations of biochemistry, gene expression, behavior, neuropathology, layered with different types of imaging analysis. The second key aspect of the platform are the in vivo animal models that we use that include wild-type animals for pharmacokinetic and target engagement studies. We conduct in-house evaluations of transgenic models of Parkinson's disease and ALS, and we access additional models through collaborations and partnerships. Again, these are used for evaluations of target engagement, model characterization. Is this a model that we think would be useful for our program? In vivo target validation, chronic studies for proof of concept, for preclinical proof of concept, and early biomarker development. And finally, in addition to our cross-functional interactions with our Neuropore colleagues in chemistry and in vitro pharmacology, the in vivo group uses collaborations and CRO partnerships where we manage outsourced ADME and PK and efficacy studies with industry and academic partners. We utilize a CRO vivarium. And again, we use specialized providers like Redoxis to access models. It's just a much more efficient way to conduct our first approximation studies. So how are we using the Luminex platform? We're using it in biochemical and gene expression measurements using singleplex and or multiplex kits to develop and validate in vivo models, to use them for in vivo target engagement profiling, for characterization of preclinical mouse models, for efficacy evaluations in preclinical mouse models, looking for any signal of benefit in those models, and for early translational biomarker methods. And what I mean by translation in the context of our drug discovery efforts, we look for translation from in vitro to in vivo and as close as we can to human and back and forth. So an iterative process so that we can learn and adjust. So now I'm going to shift gears to describe the program that we're very excited about, our TLR program. Now, toll-like receptors are host pattern recognition receptors positioned to play a role both in the innate and adaptive immune responses, and their activation triggers cellular inflammatory responses to molecular patterns. Now, I'm going to refer to them as TLRs. TLRs are localized to cell surfaces and intracellularly. They can be triggered by pattern-associated molecular patterns, which we refer to as PAMPs, or damage-associated molecular patterns, DAMPs, with MyD88 dependent and independent signaling leading to inflammatory signaling. Now, PAMPs can be thought of as non-self, such as viruses and bacteria, and DAMPs can be thought of as self. In infectious diseases, TLR activation initiates an immune response that results in elimination of pathogens, and that's usually followed by resolution of an inflammatory state. In neurodegeneration, however, we have evidence that the activation of these receptors exacerbates neuronal damage and further release of DAMPs, promoting a persistent inflammatory state without resolution. So this is a chronic and maladaptive state. Now, the program was initially focused on the hypothesis that aberrant TLR2 activation in glial cells and or neurons plays key roles in the processes underlying neurodegeneration, primarily Parkinson's disease was our initial focus, where we know that TLR2 is upregulated in patient samples from Parkinson's disease patients, as well as other neurodegenerative disorders, and this correlates with misfolded protein burden. If you activate TLR2 in animal models, you increase misfolded protein and pathology. And if you block this in vitro, you can basically you can ameliorate this. So it's that we have evidence that TLR2 downregulation reduces protein pathology and blocks neurodegeneration. So our thought process is that our application of antagonists as a therapeutic intervention may provide benefit by two complementary mechanisms: by blocking chronic activation of glia to reduce neurotoxic inflammation, or by reducing the block of protein clearance mechanisms in neurons, resulting in reduced protein aggregation as you restore their clearance mechanisms. Now, Neuropore's TLR2 program in vitro platform was built to guide compound selection for advancement. The in vitro platform development and early screening was supported by the Michael J. Fox Foundation, and the in vitro team employed multiple cellular systems as shown and readouts for compound screening and characterization. They set very clear decision-driving criteria and multiple strategies targeting the extracellular and intracellular sites of the TLR2 receptor were targeted. And this platform was used to evaluate over 62,000 compounds. And from this campaign, several novel and nanomolar potent and selective receptor antagonists were identified. So here I'm showing you our screening tiers. So that's basically our strategy of how we're going to prosecute the evaluation in the drug discovery process of our compounds. And so on the left, you have very clear go and no-go drivers for our decision-making process with regard to in vitro and in vivo evaluations. And by design, the screening tier is indication agnostic. Basically, we use the criteria on the left to learn about our compound and what it does in vitro and in vivo. And then based on our findings, we decide to conduct another set of informing endpoint studies that may be more tailored towards specific indications. And so this allows us to be opportunistic in terms of explorations of indications. So screening in recombinant TLR2 hex cells identified a compound designated as NPT 1220-312. And I'll refer to that interchangeably as that or 312. So this compound exhibited sub-nanomolar potency with minimal cellular toxicity, and outsourced ADME and PK evaluations didn't reveal any issues with the compound. So that's, and then shifting a little bit in terms of our informing endpoints, we being the in vitro group, found that our compound blocked TLR2 signaling and cytokine release of a particular cytokine, IL-8, in peripheral and central immune cells. It blocked NLRP inflammatory markers in differentiated human macrophages, and it blocks biologically relevant activators of TLR2 signaling, such as P. acnes and P. gingivalis. So in parallel, the in vivo pharmacology group was exploring multiple options for in vivo target engagement. And what I'm showing here is based on a developed and validated acute peripheral TLR2 agonist-evoked model that's conducted in wild-type mice. And we use the PAM2 agonist, PAM3, to evoke a cytokine response, and we validated that which of those analytes are sensitive to TLR2 inhibition. So using a 32-plex plasma kit, a 32-plex mouse kit in our samples, we evaluated the evoked plasma profile using the Luminex platform. And I'm showing here on the upper panel and lower panels the cytokine concentration response curves. So we also have the compound exposures from plasma. And so the upper graphs show data for the two of three mouse homologs for IL-8. So if you recall, that's the analyte that the in vitro group evaluated, demonstrating that we have a dose response, concentration response curves here of our compound versus TLR2 activation. And on the bottom, I'm showing two highlights of analytes that show IL-6 and MIP-1 beta that are relevant to neurodegeneration. If you look at the literature, those pop up pretty frequently. But if you look across all of the analytes, the overall response profile demonstrates nanomolar potency of about 550 nanomolar of our compound across a broad panel of analytes as measured here. And again, this is in wild-type mice. So, you know, I've talked about the overall response profile, and you know, I'm showing here basically some exploratory PCA modeling as one way to visualize this change, this normalization of the profile. And so on the left, the largest circle is basically the non-evoked state. So you have the animal gets a vehicle pretreatment, they get a vehicle treatment, and that's basically the boundary of their response profile. That's the larger on the left. On the right is the TLR2 agonist-evoked profile. So you see it's shifted to the right. Now, this profile, if you look across analytes, it's robust, it's complex. I've referred to these as cytokines, but it's a mixture of cytokines, chemokines, and different growth factors. So we're getting a pretty reliable and robust TLR2 agonist-evoked profile. And our treatment with TLR2 antibodies or our compound normalized this response, shifted back left towards the vehicle group profile. So this is just a visualization of how useful it can be to look at multiple analytes rather than just focusing on those that you go into based on the literature. So in the background, the Vitro Pharmacology group added additional TLR subtype evaluations because we needed to know how specific this was to TLR2. And they determined that our program molecules actually have similar potency at TLR and TLR2 and TLR9 receptors. Again, going through the list, they block both TLR2 and TLR9 activation of peripheral and central immune cells. They block synthetic and natural TLR activators, and they block NLRP inflammasome markers in differentiated human macrophages. And like TLR2, there's evidence for TLR9 involvement in multiple neurodegenerative diseases. TLR9 is an intracellular toll-like receptor, and TLR9 activation releases mitochondrial DNA that leads to immune cell activation, including activation of microglia in the CNS. So from the perspective of the link to inflammation and microglia, you could see how this would be possibly relevant. So using the Luminex platform, we've conducted multiplex evaluations of plasma cytokines. Again, in the top panel, we have the in vivo TLR2 engagement data, some of that for the mouse islate homologs with one of our newer program molecules. And so basically, that's tracked very well with our early findings with other program molecules. And in the bottom panels, these are related to our development of a TLR9 target engagement, in vivo target engagement model for program molecule evaluations. Again, demonstrating that we could block TLR9 agonist-evoked cytokines and a cytokine profile if we pretreat with our compound. So overall, the multiplex evaluations of plasma cytokines have really enabled us to make unbiased comparisons of the response profiles of our program molecules. And again, that's something kind of important as we move into the space of trying to decide how we want to evaluate these with respect to different models of indications. So with that in mind, our expertise really lies in Parkinson's disease, but we took a step back and decided to do an indication. We conducted an indication exercise. We evaluated over 60 CNS and non-CNS indications, sort of to give us a feel for what the potential of this program might be. All of the indications have a pathogenic inflammatory component, plus evidence for involvement of TLR2 and or TLR9 as derived from studies of patients or preclinical models. There are many opportunities here, as you can see, not just in the CNS space, to diversify applications in large or rare patient populations with unmet need, which is a key criteria for us internally. And importantly, TLR9 and TLR2 inhibitors have advanced through phase 1 clinical safety evaluations without any issues to date. So ultimately, though being the small company, we had to focus a bit, and so we decided on 3 key indications for our informing endpoint studies: Parkinson's disease, ALS, and Guillain-Barré syndrome. So very briefly, some of you may be familiar or likely familiar with Parkinson's disease. Again, it's a disease that has a protein pathology. Alpha-synuclein protein pathology is a hallmark of Parkinson's disease. Neurodegeneration and neurotransmitter changes lead to network dysfunction that are manifested by dynamic changes and motor dysfunction and non-motor dysfunction features. These are accompanied by neuroinflammation, impaired protein clearance mechanisms, and mitochondrial dysfunction. Now, there are familial and what we would call sporadic. So those are forms of Parkinson's disease where there's no known genetic component. So they're familial and non-familial forms of Parkinson's disease. It has a prevalence of 100 to 200 in 100,000, and so it's not actually considered a rare disorder. It has an average age at onset of about 60 years, affects men slightly more, and patients live typically for about 10 to 20 years after diagnosis. Major contributors, factors contributing to mortality are falls and pneumonia. And there are scientific rationale for the application of TLR2 and 9 antagonists for the treatment of Parkinson's disease. Here is just an overview of findings in patients indicating that, or showing that TLR2 receptors and TLR9 receptors are increased in Parkinson's disease patients' brains. So these are post-mortem evaluations. What they also know, because they have the accompanying clinical data, is that TLR2 expression correlates with increased alpha-synuclein burden and disease duration in these patients. Okay. We also have evidence from animal studies and basically confirming the roles of these two receptors in a neurodegenerative-like process. And so combinations of knockout animals and or antisense inhibitory applications with existing familial and or toxin-induced models shows us that you can reduce neurodegeneration by blocking these two receptors in various animal models. For ALS, again, we have a protein pathology component, and that's leads to progressive degeneration of corticospinal tracts and ultimately muscle loss, which is then manifested as symptoms of where you see progressive loss of muscle innervation, leading to dysfunction of voluntary movement and ultimately respiratory complications leading to mortality. There is a strong inflammatory component that's been noted in ALS, and there's a potentially clinically translatable marker of TSPO PET, which is a surrogate marker for inflammation for ALS. That's been well characterized. For clinical subtypes, again, there are genetic forms as well as sporadic forms, as well as different variations based on the limbs, the progression of whether or not it's arms versus legs, how that progresses. Patients typically are younger at onset. This disease is much more rapid in terms of progression. Males and females are equally affected. And unlike Parkinson's disease, ALS is considered a rare disorder with a prevalence of 5 in 100,000. And again, we have a whole host of scientific rationale for involvement of these two receptors, toll-like receptors 2 and 9 in this disease. And we have evidence from our own internal programs demonstrating that if we antagonize these receptors in the mouse model, that we can improve multiple endpoints potentially relevant to the disease. And then finally, Guillain-Barré Syndrome. This is a little bit different in that this is a acute demyelinating polyneuropathy. And its disease presentation is a little bit different. To be honest with you, quite a few of these patients recover, but there is a substantial number of those who don't. This is considered a rare disorder, and it's not typically that doesn't typically lead to death, but it does affect the patients. And there are some subset of patients who actually never fully recover from this disorder. And again, we have rationale for why we think this approach, the antagonism of TLR2 and TLR9 receptors, might work as a therapeutic strategy for Guillain-Barré syndrome. And I won't go through that all the way, but I will tell you that we have a little bit of data from evaluations of these dual inhibitors in preclinical animal models of Parkinson's disease, ALS, and Guillain-Barré syndrome. And altogether, these results demonstrate that this intervention improved multiple disease-relevant outcomes in all three models. So in Parkinson's disease and ALS models, there were reductions in protein pathology, inflammatory markers, as well as improved motor function and health. And in an outsourced study of the rat EAN model of Guillain-Barré syndrome, we saw improvements in health, disease severity scoring, and a reduction in improvement in myelination. So now I'll share, for the last bit of my talk, I'll share the highlights of our findings to date for our findings in the Parkinson's and ALS models that were enabled by our use of the Luminex platform. So first, these are data from a 1-month evaluation of our early program molecule, NPT 1220-312. And the take-home message here is that treatment for 1 month with our compound improved a cytokine profile in a transgenic mouse model of Parkinson's disease. So with what you see here are, let's see, the smaller circle that looks like it's on its side, the oval that's on its side, is the non-transgenic vehicle. The more upright circle on your left, that would be the transgenic model that's just vehicle treated. And then you see in the larger circle that's sort of on its side, that those animals have a what could be interpreted as a normalized profile. And so this is very interesting to us. And, we scratched our head a little bit because there's a lot to unpack here in terms of, a normal cytokine profile in an animal model and what we do to it with our treatment. But we're working on that. So on the next slide, we have data from our first evaluation of the same compound in a transgenic mouse model of ALS. And, we administered an early program molecule, again, the 312 compound, to mice, these SOD1 transgenic mice, for 1 month. And so after we finished the life portion of the study, one of our in vivo scientists identified and evaluated available study samples, which unfortunately were brain, but not spinal cord, because we had allocated the spinal cord in its entirety to neuropathology evaluations. So we didn't have any for biochemistry or this gene expression. But one of our group took this brain samples and used a quantigene multiplex kit on the Luminex platform and looked for changes or differences in TLR specific gene expression and TSPA, which if you recall, that's the marker, a surrogate marker for neuroinflammation that's been evaluated in ALS. And so on the left, you see just the simple bar graphs. And as you can see, the differences between the left two bars, those are the non-transgenic and transgenic control groups. They're not huge differences. But there were suggestions that we had some differences between the non-transgenic and the transgenic, as well as possibly some treatment effects. So if we looked at the profile of this gene expression for this kit. And you could see what basically this visualizes what I just described to you, which is that you've basically got an overlay of all your groups, and here and there it looks like you have hints of separation, possibly for treatment. So basically we decided that because it was such an easy evaluation to run, that we would allocate some sample in our next study specifically for this purpose. And so I'll show you this now. So here, these are data from the next and most recent evaluation. This is a study of a newer compound of 1220-478. This is an ALS Association funded study. This was done again in the SOD1 G93 transgenic mouse model of ALS. And so again, in our portion of reserved spinal cord, our researcher evaluated the samples across this platform using this technology. And this time, being we're in the spinal cord, which is the focus of most of the most severe pathology in ALS, this time we found very, very robust increases in expression of these various TLR receptor subtypes and TSPO. And moreover, we also noted that we had what looked like treatment effects in these same samples. And if you look across, it looks like the most consistent effects are at the 10 mg per kg. So we went ahead and decided to visualize that here. And so what's shown here is a radar plot of this abnormal TLR and TSPO profile. And so in the center, you've got the dashed lines. So that's your non-transgenic control. The outer bounded filament here is the transgenic vehicle controls, and the intermediate line is the 10 mg per kg treated group. And so this is a nice way to visualize where the changes are. And we're very happy with this. Maybe not the most straightforward set of data. Some people look at this and they say, "Oh my goodness, but TLR7 has moved, and you're a TLR2 and TLR9 antagonist. How is it doing that?" Well, it's you're looking at the sum of what's happened as a result of your chronic administration. So there's the possibility of compensatory changes as well, and also that crosstalk between different pathways. So we're not bothered by that at all. Okay. So finally, I think I've got a few more minutes. In the last few slides, I want to share data from a translational biomarker development effort that again uses multiplex kits on the Luminex platform. And so we have been working on developing and evaluating TLR agonist-evoked whole blood cytokine assay experiments. So this is a method where you can obtain, in this case, mouse or human whole blood. And we have access to both in-house for the mouse, and for human, we've at this point in time mostly purchased the blood from a blood bank. But you could imagine down the line, we could do the same thing with evoked models for the mouse. You could take normal, healthy, or patient blood, and then in vitro, which is the next step, you can apply your intervention and your agonist to see evaluate not only your difference between your groups of interest, but also a response profile, which is actually key, in my opinion. And then, again, using that on employing the Luminex platform. So this isn't something that we came up with on our own necessarily. This is a method that's already been applied in phase 1 and 2 studies as measures of target engagement and for proof of mechanism biomarkers. So that's something that someone has already successfully done, not for neurodegenerative diseases, but for another modality. And in this case, we're really excited about this because we think this could be important in terms of a patient stratification biomarker. So remember, I mentioned that sometimes you have non-responders to your treatment, and those those are part of your data analysis. You can't just make them go away. But if you knew that they're non-responders ahead of time, you might be able to basically use that as an inclusion-exclusion criteria. So by doing that, you can decrease your variability that's being contributed by that. And so fit for purpose variations in subject, treatment condition, in vivo versus in vitro treatment, et cetera, can be used to address specific questions. And so we've started to do this in the context of the ALS program. But first, let me just show you the data from our method development of wild type and normal healthy humans. And so here I'm showing you the TLR2 mouse whole blood cytokine assay data on the top. And the important thing here is that in running these evaluations where you run your agonist response curves, you find your effective concentration 50%, and then you basically run your compound dose response versus that in a separate experiment. And so what we learned was that this method translates really well and lines up with what we had found in terms of our in vivo and in vitro potencies. So we're very, very happy with that. And that really is helpful as you start to project what your therapeutic doses might be for the clinic. So that's for TLR2. For TLR9, we also have developed and validated that for mouse blood. And we've transferred both the TLR2 and TLR9 assays for use with normal human blood. And we like this, one, because the multiplex evaluations give us a very good handle on the response profile across species and within subject. So we decided for my final slide, almost final slide here, my final data slide, I'm showing you data from an evaluation where we had a satellite group from an experiment in the SOD1 ALS model. So we treated these animals for 1 month with our compound. And at the end of the experiment, the study, we took this group of animals and at a PK-defined and time point past their last injection, we took our sample, and then we used that blood. So this is an in vivo treatment, and then we use that blood in vitro to conduct our agonist response curves, just to see what those response profiles looked like between the groups and across the analytes. And so this first pass evaluation, actually, I think was pretty interesting. We have some example data here below where you see we have a GCSF here on the left, and it looks like our control groups are pretty much laid over each other, but we have some evidence that our treatment intervention, and that's the curve on the bottom, gives us some evidence that maybe this particular analyte is a possible treatment responsive analyte. And the same thing for our IL-8 analyte. And then on the far right, we have IL-6. And in this case, it looks like we have possibly a starting difference in levels with this analyte, which is something we'd actually seen by another technology previously. And we see a different response profile. And that we think is really interesting because it gives us another way to differentiate, possibly to differentiate patients. Because, for instance, in Parkinson's disease, we know that not only are basal levels different, but the function of the integrity of the inflammatory response or the immune response is actually changed. So in this case, that really lines up with what we see here. And so we have more to do here with respect to ALS, and we're also planning to extend these evaluations to our Parkinson's related evaluations. So my last 30 seconds. In summary, the discovery and development of CNS disease-modifying therapeutics for neurodegenerative diseases is a difficult process that will improve as the complexities of the disease are elucidated. So we need to learn more about the patient and the disease. The development of biomarkers is a key part of that process, and I and many other people, most of the people in the field believe that has to be done early. And I hope that I've given you some indication and data showing you how we've been able to use the Luminex platform as a key technology for this process and for our early translational biomarker development efforts. Thank you so much.
All right, thank you so much, Dr. Price, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen. Now let's get started. Our first question, so you showed TLR gene expression data. What about biochemical evaluations?
Thank you for that question. We are very interested in doing the biochemical evaluations, and I mentioned that companies like Luminex and Neuropore and Thermo Fisher Scientific, those are all what we consider industry partners, in that we contact people and we say, this is what we're interested in doing. How do we build a kit for this? And they don't get paid. They help us out. They've been helping us out. So we are building assays for evaluation of biochemical evaluations. Our only limitation are basically just what reagents are available to help us with that.
Great, thank you. Another question we have, don't we already know which plasma cytokines are different in Parkinson's disease patients?
We know part of that. So when I've mentioned the term unbiased, part of the issue is that as a field, we've been limited by our technology and our reagents. And so researchers use what they have available to them. And so previously, kind of the best that we could do on many levels was just singleplex evaluations. And they often weren't, they were more qualitative than quantitative. So now with the emergence of technologies like the multiplex kits and Luminex and other available technologies in the field, we can kind of broaden our approach now, and I think that's actually really helpful. It's like trying to decide how a room looks by looking through the keyhole. You don't really know what it looks like until you open the whole door, and that's really what we're doing here by using these technologies, is opening the door so we have a better view of what's inside.
Thank you for that response. Another question. I noticed that you didn't mention the role of animal models in translational failures. What part do they play?
Thank you for that question. That's a great question and a totally different talk, but I will say, as a person who's used animal models throughout my career, I mean, I have over the last 30 years, watched as animal models have taken the brunt of the blame for translational failures. And there's a fair amount to be blame to be had with how we as researchers have used those models. Over interpretations, inappropriate applications. But ultimately, without that information about the patients that I talked about, it's really hard to know how good your animal model is. And in the end, it really matters what your questions are specific to your therapeutic intervention and what your target is. So it's complicated, but that's a good question. There's blame to be had there, but ultimately, without that other information, you can't properly apply and use animal models. Thank you.
Thank you for that. Another question we have from the audience. What are some of the immune responses produced from the TLR2 and 9 antagonists? And do these antagonists recognize the multitude of immune responses necessary to contribute to your research goals?
That's a good question. So in this case, what we've done, Like, this is a little bit of a complex question, but for each of our agonist-provoked models, the way we validate it is with available tool compounds, not our own, to basically look across that profile and say, of the 32 analytes, which of those could we measure? Because sometimes you have some that are consistently below the limit of detection. And of those, which of those are sensitive to the inhibition using the best tool that we have to block them? And so it's not perfect, but again, it tells you how important it is to look at an entire profile. So, that allows us to see which analytes are sensitive. And by sensitive, I mean they have to be, you have to have a pretty good response to the antagonism in terms of your blockade. So, over 50%. And there's overlap, so it's complicated. That's a good question. Thank you.
Thank you. We have some great questions coming in. Another one, what is the relative importance of neurobiomarkers versus systemic biomarkers? Example, inflammation related.
That's a great question. And this is another one of those, it's complicated answers. So ideally, we would have a way to directly measure inflammation in the brain. In our mouse studies, a lot of what we do post-mortem is basically what I would call like snapshot evaluations of inflammatory markers in the brain, either by using immunohistochemistry or biochemical techniques that we just can't do with patients, it turns out. So we're really limited there in making that jump. But we do know that in some way what you see in the periphery is often a reflection of what you see centrally. And so that is actually a very, very important part of the process for us. And one that we're working through as best as we can. I primarily work with mice, and so, bridging from the periphery to the brain, it might be nice to be able to do a CSF evaluation. It's really hard to find in vivo pharmacologists who can do a very, very good CSF draw on a mouse. It's really hard. So we're doing the best we can. We have some ideas of how we might actually answer that a different way, but that's a really good question.
Great. The next question we have, do you see changes in the same cytokines across different neurodegenerative disease models?
That's a great question. Thank you, Carrie. That sometimes we do. Sometimes we don't. And it also depends on, I briefly mentioned a treatment intervention point for patients, and that actually complicates our use of the animal models quite a bit, because there's a lot that goes into deciding how old the animals need to be. At what point in their lifespan do you use them, in terms of, do you have developed pathology that's accompanied by symptoms? So let's see. I lost that question. I'm sorry.
That's okay.
Yeah. So that changes. So it depends on what point of the animal progression for each line, which animal model. So yes, sometimes there's overlap. It's not always easy to interpret. There is.
Great. Another question we have, what made you decide to use the Luminex platform versus other available platforms?
Good question. I had a very talented biochemist in the in vivo group who noticed the platform and made a really compelling case to me and the management. This is why you need it, because it's we were struggling using the mice in terms of, you have sample from brain or plasma, and if you're going to run a Western blot, it takes a huge amount of sample, and then you're done. You basically, that's it. That's the last evaluation that you'll have. And so, she made the case that, given the amount that the system costs and the kits, it would allow us to be very, very flexible we wouldn't have to use very much sample. And so, in the end, she did a very good job selling it. And that's basically why we decided to invest in the platform. And then we convinced our in vitro counterparts that it was also a good buy. So.
Great. We have time for just a couple more questions. And the next one we have here, aren't multiplex studies really just fishing expeditions?
That's a good question. Yes, in one way they are. There are a lot of endpoints for clinical evaluations that really just haven't panned out. And so part of what the field's doing in general is taking a step back and saying, hey, maybe we need to think outside the box a little bit here. And so I think it follows that if there's comfort in the clinical space in doing that in early clinical trials, that preclinically in animal models, we should be kind of following suit, because we don't always know what's going to be important. And so, it's funny thinking about genetic, the genome evaluations. People have gotten very, very comfortable in evaluating tens of thousands of genes. And essentially that would be a fishing expedition. But yeah, I think there's just a level of comfort now that the technologies have advanced to enable that type of evaluations. There's just, there's a time for the rest of the field to catch up and become comfortable with the types of analyses that go with that and using it in a way that's proper. So, yeah.
Great. Thank you, Dr. Price. I think we are going to wrap with that final question. Do you have any final comments for our audience?
Not too much. Thank you for coming today. And I'd be happy if anybody wants to ask me something that occurs to you later offline. Just email me and I'd be happy to respond to you.
Great. Thank you again, Dr. Price, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsor, Luminex, a DSORN company, for sponsoring today's webinar. And like Dr. Price said, questions we do not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until March 10th, 2024. LabRoots will alert you via email when it's available for the replay. We encourage you to share that email with any of your colleagues who may have missed today's live event. Until next time, goodbye.